We have before us a highly relevant scientific study that proves a drug potentized above Avogadro limit or above 12C can antidote the biological effects of same drug in crude or molecular forms, as predicted by MIT explanation of scientific homeopathy. This study was conducted by a team headed by Anisa Rahman Kudarbaksh of Department of Zoology, University of Kalyani, Kalyani 741235, West Bengal, India, in association with Boiron Lab, 20 Rue de la Libération, Saint-Foy-les-Lyons, France, and published on December 26, 2005. Complete report is available at this link, http://www.ncbi.nlm.ni. Gov PMC Articles PMC 1375236. This team undertook a study to find out whether administration of potentized homeopathic remedy, Arsenicum album, alter anti-nuclear antibody anatiter in people living in high-risk arsenic-contaminated areas. To examine whether elevated anti-nuclear antibody anatiters reported in random human population of arsenic-contaminated villages can be reverted to the normal range by administration of a potentized homeopathic drug, Arsenicum album. Randomly selected volunteers in 2 arsenic-contaminated villages and 1 arsenic-free village in West Bengal, India were periodically tested for their anatiter as well as various blood parameters in 2 types of experiments. Placebo-controlled double-blind, experiment for shorter duration and uncontrolled verum-fed experiment, for longer duration. Positive modulation of anatiter was observed along with changes in certain relevant hematological parameters, namely total count of red blood cells and white blood cells, packed cell volume, hemoglobin content, erythrocyte sedimentation rate and blood sugar level, mostly within 2 months of drug administration. This controlled scientific study has sufficiently proved beyond that potentized homeopathic medicines can antidote or reverse the biological effects of parent drugs. In the absence of original drug molecules, how could the homeopathic potencies exhibit such an action? The theory that potentized medicines mimic the parent drugs is obviously disproved through these experiments. Only logical explanation we can provide for this phenomenon is the molecular imprints of parent drug molecules being the active principles of potentized medicines. Molecular imprints can specifically bind to the parent molecules, and thereby antidote or reverse the biological properties of parent molecules.